Okay, so as what I said before, just what I said just now, we will we will learn the principle again for training. Okay, I think you can see my screen, right?
Yes.
Okay, so
Flow cytometry is used to count and analyze the size, shape, and properties of individuals.
can you hear the voice of the video?
Yes, we can hear.
Okay.
Flow cytometry is used to count and analyze the
Sorry,
size, shape, and
I have another version that have the English subtitle here.
Flow cytometry is used to count and analyze the size, shape, and properties of individual cells within a heterogeneous population of cells. Flow cytometry data is extremely quantitative and can be analyzed in depth by specific flow cytometry software programs. Once a suspension of individual cells has been prepared for flow cytometry analysis from cells, tissues, or organisms, the prepared sample is placed on the flow cytometer. How does a flow cytometer analyze the size, shape, and properties of individual cells? A flow cytometer contains several key components, including the sample, fluidics that move the sample into the flow cytometer, lasers, optics which gather the light, detectors to sense the light, and a computer system to output the data into a form that can be analyzed by the researcher. Once a sample has been placed in the flow cytometer, the machine will suck up the sample, mix the sample into a stream of saline solution in the cytometer, and lead the cell suspension through a narrowing channel, causing the cells to form a single file line before the cells pass through the laser at the interrogation point. Each cell within the sample then moves across the laser beam, allowing each cell within the sample to be individually analyzed. As each cell passes through the laser beam will scatter in multiple directions. The flow cytometer detects light scattered in a forward manner, called forward scatter, and light scattered in a sideways manner, called side scatter. The amount of forward scattered light for each cell is detected by a detector on the far side of the cell from the laser. Forward scatter is proportional to the size of the cell. The detector converts the scattered light into a voltage pulse, which is directly proportional to the amount of forward scattered light. The computer converts these data into a histogram plot with the amount of forward scattered light on the x-axis and the number of cells on the y-axis. The amount of side scattered light is detected by a detector located perpendicular to the path of the laser beam. Side scatter is proportional to the shape and internal complexity of a cell. Just like with forward scattered light, the flow cytometer converts the detected side scattered light into a voltage pulse, which is directly proportional to the amount of side scattered light. By analyzing the forward and side scatter data together, the researcher can understand a cell's size, shape, and complexity. In addition, the analysis of the forward and side scatter data can allow the researcher to divide the heterogeneous population of cells into individual populations with varying size, shape, and complexity. This capability of analyzing multiple populations within a sample is the immense strength of the flow cytometry experimental technique. In addition to analyzing a cell's size, shape, and complexity, a flow cytometer can also detect emitted light from excited fluorescent molecules, such as fluorescently labeled antibodies or fluorescent dyes or stains. For the rest of this animation, we will focus on the detection of fluorescent signal by the flow cytometer. For more information about fluorescence, please see the fluorescence animation. The fluorescent molecules, or fluorophores, are excited by the correct wavelength of laser light when the cell passes through the laser beam. After excitation, the light emitted by the fluorophore is directed along a path with emission filters that allow for the detection of multiple different fluorophores emitting light in a cell. A flow cytometer can detect the presence of a fluorophore and also quantify the relative amount of a fluorophore within a cell. A flow cytometer can also be used to detect light emitted from multiple fluorophores in the same sample. While it is common for flow cytometers to detect 2, 3, or 4 colors at the same time, some flow cytometers can even detect up to 18 different colors at the same time. The data can then be presented to the researcher in the form of a histogram or a dot plot. Such data furthers the powers of flow cytometry analysis by enabling the researcher to analyze individual populations of cells within a heterogeneous mix of cells in the sample.
Okay, I think the video is very clear. Uh, Let me check. Okay, do you guys have any problem or which parts you think that is hard for you? And because now I will introduce the principles more in details, more specifically. Okay, if no one's uh, answering, so okay. Now let's learn this principle in details, okay? And I will make it quick, make this introduction quick. Just as the flow tree's name. Okay. Flow means to move in a stream. You see in the video, we see the single cells flow as a stream. And the settlement cell, because the object be detected always be the cells, but also you can use it to detect other things, but mostly the cells. And metry is to measure. And here is our machine. It's like this. And we know the flow cytometer is a little bit same as the hemostatometer, right? Because they both have lasers and channels is like that. So and the result is actually this basically the same. Use the same principle here. But I think you already know the hemostatometer well, so I'd like to uh, introduce the differences between the two machines now. We know the hemostatometer use blood to detect directly, actually. But the flow cytometer, we have a step or a procedure outside the flow cytometer here. Outside the flow cytometer, it's like we have to stain the cells. What we use to stain the cells? one is the fluorescence. It's just like the dyes. And then another one is antibodies. So what is antibodies? It's like a key. It's like a key that specifically bind to the lock. The lock is expressed on the cells. It may be some proteins or some lipid. I don't know how to say sorry. It's like some proteins or some it's like that. After the binding, the cells will be bind by the antibodies, and we have some ways to detect the antibodies we used. So after the detection, we can get the information of the antibodies, and then through analyzing the antibodies, we can get the information of the cells. And here's the review of the 6 series hematology analyzer. It's like 6180. It's like that. So we can see in the hematology analyzer, we do have a laser like this here. And then we also detect the FS and the SS and also the FL. It's just like so you see, we use the laser here to shine the cells here. So in the front uh, angle or the zero angle, here's the detector, and this is the FS. It's like this is the FS, just like this FS, and then here triangle level, but sorry, angle here will be the SS and FL. It's like that. And in the 6 series hematology analyzer, we already use the fluorescence dyes to detect the DNA and RNA. You see here, we use the dyes to stain the DNA, RNA in the cell. So after the detection like this, through the FL Psi fluorescence, the FL, we can get the information of the DNA and RNA. So flow cytometer's optical system is more complex, but the principle, the basic things are the same. So we have lasers here, but usually more than one laser. For example, we can choose to equip the machine with one laser, two laser, three laser. The maximum is three laser in the BF730. And then we can get the FSC, actually the FS information here, and also the triangle angle. So sorry, it's like the lighty angle. Here we can get the SSC here. And we use a series of obstacles here and the filter here is like the filter to make to separate the light or separate the fluorescence here, and then define the separated lights or signals here as the, for example, FITC and PEAPC and so on. So the differences between the hematology analyzer and the flow cytometer is like the optical system are more complex because it can equipped with several lasers, and the channels are more. So for example, we can have the FITC and P and APC. Each channel can get one information about the cell. For example, we know in the 6-series hematology analyzer, we can use one because it only has one side fluorescence channels, and use this to stain to detect the DNA and RNA information. But for flow cytometer, we can choose which fluorescence channels to detect the DNA RNA if you need to get the information of the DNA RNA content. For example, we can stain the cells with the fluorescence that have the fluorochrome of FITC, and then after detection, we can get the DNA RNA content by the FITC channel. And if we want decide to use the PECV7 channel to detect the DNA RNA content, we can use the corresponding fluorochrome corresponding dyes to stain the cells and then get the information by the PECV7 channel. So we see so let's figure out the definition first. So well, all in the flow cytometer, we always say you know the lasers and the channels. So how we note the lasers down, not that how we express the lasers or the abbreviation of the lasers is like, for example, in this picture, we use two lasers here. One is, you know, here the blue laser, the wavelength is 488 nanometer here. And also we have a red laser here, which the wavelength is 635 nanometer. So in this picture or in this specification configuration, we have two lasers and we name the two lasers as B and R because the abbreviation blue and red, like that. So here's another item, the channels. So we see here, one channel FITC here, and then another fluorescence channel PE, APC is like this. So in the following PowerPoint, we will learn that one laser will have its own corresponding channels. And for example, in this, let me directly tell you that we have three channels can be excitated by the blue laser. So and one channel that can be excitated by the red laser. So in this configuration, we will name this as the B3 and R1. It's like that. And here's another comparison. You see, in the 6-series hematology analyzer, we only use fluorescence dyes to stain the DNA and RNA. But in flow cytometer, we can choose because we have so many channels, right? The maximum is 14. So we have so many channels, so we can detect which channel to stain the cells to guess which information we need. For example, if we want to know the DNA content, just like this, because we have 14 channels, we can choose which channel to detect the DNA content, and then we use the corresponding reagent to stain the cell. And for example, if we want to get other information, for example, the CD3, we can use another channel and the corresponding reagents to stain the cells, to stain the CD3. And after detection, we can get the information of the cell size because the FL and complexity is like at the FS, and the cell surface things and the cell interior things because we use the corresponding reagents. And the related terms is like, the white blood cells have these, and the DNA and RNA is like this is in the nucleus. And how we stain the DNA is like this purple one is the stain, the dyes, fluorescence dyes, and bind like this way. And we learned we have, three lasers. In the BF730, we have 3 lasers and 14 channels. So how can we get the information of the different channels? So it's like we use a reagent, the reagent called the fluorochrome here. So let's see how it detects the fluorochrome to get the information of the cell.
Is a functional molecule that fluoresces or emits light. How does a fluorochrome fluoresce? When a fluorochrome is excited by an energy source, such as a laser beam, the fluorochrome absorbs the energy and then emits energy of a higher wavelength. For example, when a FITC fluorochrome is excited by a 488 nanometer laser, emission of wavelengths at a...
You see here, the excitation is the laser beam. We can use a blue laser is 488 nanometer and the red lasers. And this is the reagent called fluorochrome. And the fluorochrome will absorb the energy of the laser and emits the lights or the energy here.
Approximately 520 nanometers occur.
And just like this is the excitation, is the laser here. Oh, do not care about the color because this two does not match. And this is the energy of the excitation. And here shows the energy loss in the procedure when the fluorochrome absorb the excitation and emits the emission here. So it's the energy loss. And then there's the emission light like this. So the excitation here and the emission here, this is the energy loss. And because we learned the physical knowledge in the study period, in our study period, so we know the higher the energy, the shorter the wavelength of the light, right? So here, the excitation get higher energy, so the wavelength is smaller here. For example, here, and there's the energy loss, and this one is the emission here. So emission is longer. The wavelength is longer.
Is a functional...
So it's like this. The excitation, this is the wavelength. The excitation like here, and the emission is like this. So here we see we use the 488. This is the excitation. In the flow cytometer, this will be the laser. So we see this is the spectrum of the excitation that can be used by this channel or this fluorochrome, this reagent. And we see in the 488 here, this laser gets the highest efficiency. So if we want to detect this precipitous 5.5, this fluorochrome, it's better to use this 488 nanometer wavelengths of laser like this. This also explains why we one laser have its corresponding channels or fluorochromes, because you see here every fluorochrome has its own excitation spectrum. So for example, this is precipitous 5.5. If we get another channel like FITC and we can see its spectrum, and then another channel, and we can see the excitation spectrum here will get will both have this one, the 488. So that means if we already fix the excitation or the laser here, so the corresponding channels are be fixed too, because one laser only have its corresponding channels. Okay, this is the item explaining the marker and antibody. Here's the antibody. The one reagent ingredient of the reagent, and this is the cell. The antibody will bind the some parts of the cell. Basically, this will be one protein here. We call this one that can be bind by the antibodies here. We call this as marker. So if you learn the marker like CD3, CD4, it means the things or the molecules we want to stain. And here we see in the BF730, we have the three lasers, the violet one, the blue one, and the red one. And we learned that because the excitation spectrum, so the blue one can have its corresponding channels like this, and the violet one get channels like this. So if we see for example, if we bought a flow cytometer from Urit, and how we express the configuration of the optical system, we can call it the violet 6, 1 to 6, blue 5, and red 3. So the abbreviation will be V6, B5, R3. We can look at this picture. Here's the cell. And on the cell or in the cell, just like the cell has some molecules we want to stain. So we use antibodies to bind the molecules here. The different molecules, like this one is one molecule, and see this one is another kind of molecule like this. Because we have these channels, we can have some these options, 14 options of the channels to detect. So we use antibody to bind the molecules, and how we make the machine know that the information we want to detect. So we can see on the tier of the antibody here. This is the fluorochrome. This is the little one that can be acetated by the laser here. So by detecting the laser, the fluorochrome here, and we can get the information of the molecules here, for example. If we can see this one, the antibody and the fluorochrome binds together. This one is the same as this one, and also this one. We can see 3 of them. So if this molecule is the marker we want to detect, we can get three signals from this cell, 1, 2, 3. And we will know the expression level of the molecules in the cell. So this is the filter to separate the fluorescent signals or the emissions. Long pass, longer than that can pass. Short pass, shorter than that can pass. We can see here, shorter than that. And the band pass means within this width, the lights can be passed. Oh, the SP here you see the number here means the wavelength of the lights. And this one is also another animation.
Optics subsystem consists of excitation and collection components. The excitation components include lasers, beam shaping prisms, and lenses to route the laser beams to the sample core. When particles pass through the laser interrogation points, they scatter light. Any fluorescent molecules present on the particle fluoresce. Appropriately positioned lenses collect scattered and fluorescent light. The collection components steer the scattered and fluorescent light to the appropriate detectors.
This is the filters, but these are the filters.
The detectors in the electronic subsystem convert detected light into proportional electronic signals. Electronic signals are then digitized, further processed, and sent to the computer. Data for each cell or particle is stored in the computer and plotted in various ways for further analysis.
Okay, so this is the data analyze. Uh, Actually, here we see the FSC and SSC. These two we call the channels detect the physical parameters of the cell. It's like the FSC detects the cell size, you see here. And the FSSC detects the cell complexity. So the bigger the cell, the higher the FSC signal. And the more complex the cell has, so the higher here, the higher SSC it gets. And then we have the fluorescence channels like this. Here, this is the blue laser. Blue laser has its corresponding channels like this. We use the channel specific or channel specific reagents. We can stain the cells and then detect the reagents, corresponding reagents. We can get the information of the marker. Okay, here's another animation. This is the flow chamber, and this is where you make the cells flow as the one by one. Okay. Um, After the introduction of the optical system, now we are entering the electronic systems. The electronic systems will detect the signal, the fluorescence, and then transform the fluorescent signals into the digital system signals.
Light signals are generated once cells or particles are interrogated by the laser. The light signals are converted to equivalent electronic signals and then further processed and converted to a number. The larger the signal, the higher the number. The data is stored by the computer system in a flow cytometry standard format developed by the International Society for Advancement of Cytometry. Light signals are
So it's like this. When the cells passing through the laser, it will has the curve of the signals.
lights.
See, this is the FSC channel. We see the small cells has lower electronic signal, and the large cells has higher. Same as the fluorescence channels. The more fluorochrome, the higher digital signals it will get. So this is the cell that we all already stained with the reagents, the antibodies, and on the top tier of the antibody there will be a fluorochrome. Means antibody and the fluorochrome binds together. So after the staining, we can run the samples directly onto the machine. So here's the results we get. The FSC, in the FSC channel, it has this high. The means the cell size, and the SSA it gets this high means the cell complexity, and we also have two fluorescence channel signals here. To mean we can see here we get high volume of the channel of AF 488, and then a lower one from the AF this channel. So here
Data.
Okay, it's like how we see this. This is the animation showed how the machine or the computer process the data we get. And the event means one particle. It's like one particle or one cell, like here, one cell. And you can see the row of the first row. The first row we can see for the first cell, we get this high of FSC, the cell size, and this high of cell complexity. And in the FITC channel, we get this high, and in the PE channel, we get this high. And this is the first cell information. And here we show the 3 cells, and the second cell, the same one. We see the 2 fluorescence channels get so much higher. So how we transfer this data into the figure that the operator or our human can directly understand is like this. You can see.
From the cytometer, it is saved as a list. The numerical values for each cell's measurements are saved. Data can be displayed in different plot types by plotting the numerical values. For example, in a dot plot, 2 parameters are plotted against each other. The FITC value for event 1 is 690, and the PE value is 875. The dot that represents event 1 is mapped on the plot at coordinates 690 on the FITC axis and 875 on the PE axis. The FITC value for event 2 is 39,271, and the PE value is 90,000. The dot that represents event 2 is mapped on the plot at coordinates 39,271 on the FITC axis and 90,000 on the PE axis. We can see that after many single events are graphed together, populations start to emerge. X9 thousand on the PE axis.
So we see the axis like this. The x-axis is PE and y-axis is FITC. So we can see this one is gets the lower signal compared with this one. And after gathering so many numerous signal events, we can get the information like we can get the figure like this. And okay, on the red part of this slide, we can see there are 2 figures, and let's learn the first figure here. So first thing is notice the ASICs here, FSC and SSC. So we can see that this group of cells has get lower signals of the cell size, and the cell complexity. And this group of cells gets higher, the cell size and cell complexity. So how we define this group of cells? So we have to learn the cells first. You see the lysed whole blood. It means these cells are from the blood. And the lyse means we already lysed the red blood cells, just like in the hemocytometer, we use lyse to lyse red blood cells, and the white blood cells be left. So combined with its the white blood cells character, so we can see lymphocyte has lower small in size and get lower complexity. So this group of cells is lymphocytes, and then monocytes and neutrophils. Okay, these 2 are the physical information. Actually, this is the same as hemocytometer. And now we learn the fluorescence channels. So here we get 2 channels, FITC and APC. Okay, just 2 different channels. And see here, this group of cells gets high volume, high signal in the FITC. You can see the green fluorochrome. And this group of cells get high APC, the orange one. And this group of cells gets two channel signals. So normally we define these two groups because they both have high FITC signal. We define these two group cells as FITC positive. And we see here the Q2 is FITC positive and APC positive. And also we see here this Q4, this group cells as FITC positive but APC negative. So, okay, this on the lower left corner, this group cells are FITC negative and APC negative here.
Data from the
Okay, after learned how the electronic signals worked, so let's see the real results. This is the result of the TBNK test. So why I use TBNK as an example, because in China, this one is mostly used in domestic market. So first, we see the title here. All events means all the cells from the blood or all the cells that be detected by the machine. And y-axis is SSC means the cell complexity, and the x-axis is CD45 or the per-CP 5.5 means we use the reagent have two components. One is the CD45 antibody, and we bind the antibody with this per-CP 5.5 fluorochrome. Okay, so we see here. This group of cells are the cells we cared about. So we see here it is low in the cell complexity and get high CD45 signals, which is CD45 highly positive. So this group of cells are lymphocytes here. And now we can classify the lymphocyte or gather the lymphocyte. And then the purpose of this test item is to figure out the subtypes of the lymphocytes. So we will have a further analyze of the lymphocyte. So here we see the rest figures comes from lymphocyte because we see the title is lymphocytes, means these cells are all comes from here. So we here use the SX2. The x-axis is CD3 here. So we use these cells to define the CD3 positive group of cells here. And then we skip to this figure. This figure also comes from the lymphocyte too. So we confirmed we cared here about, sorry, the group of cells we cared about here is CD4 positive lymphocyte. If you don't know the meaning of the marker, that is okay, because you can see this group of cells are FITC positive and P7 positive, which means the double positive. And we see here, we bind the CD3 antibody with the FITC fluorochrome. So we can see this group of cells are CD3 positive, and this group of also CD4 positive. So if you don't know the meaning of the marker, that is okay. You can tell the customer that we see this group of cells are CD3 positive and CD4 positive, and let the doctors find out the meaning of the marker. Okay, here, this group of cells are CD3 positive, CD3 negative, but CD16 and CD55 positive. It's like this. And we can see here are also some table here. Why we have to set the table here is based on the test item or the doctor's needs. So if the doctor say, I need the table like this, you can set the table here. And how we see, how we define a result is good or not. We see the cell population have well-defined boundaries. It's the purpose of this is to classify or to group the cells easily. And here we get the proportion, and the event means the cell count, and the proportion means proportion. So we can see the cell count and the proportion situation. For example, if you detect two healthy people's sample, and you get the results showed the cell events and the proportion are very strange, with or without the limitation of the healthy people. So you may set the parameter in the wrong way. So it means you have to fix that problem. So we now learn these test item. So how many test item we can be detected by the flow cytometer? So it's like the principle is like you can detect the markers as long as you can get the antibody, and you can find the reagent that combine the antibodies you need and the fluorochromes that you can detect in your machine. For example, if I want to detect the DNA and RNA, for example, if I want to detect the DNA content, I have to use the DNA antibody, sorry, not antibody, dyes or something combined with the DNA. And I need some fluorochrome too. So if I can find the reagent that have two components, we can stain DNA and detect by the machine. For example, if we want to detect here, we want to detect CD3 here. As long as we can find a reagent that bind the anti-CD3 antibody, and on the top tier of the antibody, or bind the antibodies, the fluorochrome that can be detected by the machine. Because we have 14 channels. So for example, we use FITC to bind this fluorochrome to bind with the antibody. So as long as you can find the reagents, and you can detect the cell markers on the machine. But in the real life, the principle is easy, but in real life, it has so many situations. For example, if the if the content of the concentration of the cell marker of the marker is too low, maybe the signal will be missed, or sometimes the you see the marker will be secreted by the cell. So we have to use other steps to make the proteins or the secretions within the cell. It's like that. So the principle is easy as long as you can find the reagents can be used on the machine and you can detect. But the real life, it has so many other situations. So okay, here's the principle here. So we have sorry, not in the following other slide, I will show you the test items that's already be detected or already matured in our system flow cytometer. So it's like this. This is the test items. And I will give you a list. And we learned how the main parts of the machine, and we learned the principle of the optical system or the digital system. And also after that, we learned how to analyze the results, how to learn the results. And to summary, I'd like to show you the whole procedure. Okay, I'd like to show you the whole procedure. Is first thing is you have to get the sample. Maybe some sample are blood, or some sample from our bone marrow, or some sample from our lung, a kidney, and so on. So then after we receive the sample, we should stain the cells. Stain the cells with reagents. The cells, the reagent has one component that can bind with the cell. It may be antibody, and another component that can be detected by the machine that will be fluorochrome. So the two components of the reagent. So here we use this reagent to stain the cell, and then use the flow cytometer to detect here. And the flow cytometer will get the data, get the data and will precise the data as the figure we can see, we can understand. And then we can set the table here like this here. Know the proportions or the cell count and so on. So let's review the three main system of the machine, the fluidic system here, to flow, yes, to make the single cell flow, and the optical system to detect the signal, electronic system to transform or convert the light signal into the digital signal, and precise the signal as the figure that we can see. Okay, I think these are the principles or the basic knowledge of the flow cytometer, just like this. Okay. There's the optical system, the lasers, and the cable to transfer the fluorescence channels, and this is the three corresponding parts that's these are the filters or to separate the not separates the uh signals of each channel here. And the data display, we have several forms of the results. The histogram, like this, the x-axis will be the channel, and then the y-axis will be the cell count. And this one we already learned. This one actually, these two are actually the same with this one. It's a different form. For example, the counter map means on each circle here, the cell counts are the same. And you see here, the largest circle has a lowest cell counts, and on the center of these, the purple one gets the highest cell counts. Just like the mountain, on the bottom of the mountain, the altitude is the lowest. And after you climb the mountain, climb, and then you finally arrive at the peak of the mountain. So the altitude of the peak of the mountain is the highest. Sorry, the altitude of the peak is the highest. And this one is actually the same. It's just highlighted the areas that gets the most of cells, the highest of cells. Okay, the configuration here we already learned. And these are the three a flow cytometer here. This is the inside look or the structure of the flow cytometer you may more care about, but these pictures I copied from the service menu. You can check it later. Okay. Here's the optical module like this. And then the tongue and so on, and the reagent sensor interface where we connect the reagent. And here's the splitting module. We split the signals here and the switch power. Okay, need we take break? Or if you have some questions, you can ask now.
For me, everything is clear.
Okay. Okay. Who is speaking? I'm sorry. So who
Martin,
answered
yeah.
my question? Okay, Martin, thank you. Okay, if you guys do not have other questions, or if you do not need any break, we can keep going on. And I already told the principle, the how we analyze the results, and I also have some, the operation videos. Okay, I'll show you two videos, and this one is the first one. And the first one is uh, concerned about the basic operation. Can you see my screen now? I think you can too.
Yes.
Okay.
Power on. Press the power button to turn on. Double-click to open the site or software. Username is admin. Password is 123456. After entering, the lower left corner will show the startup cleaning status, and the reagent status will be displayed in the lower right. After the boot is completed, the lower left corner will change to ready state. At this time, you can perform quality control tests. Quality control test. Prepare QC reagent. Take the QC reagent from the refrigerator. Confirm that the QC reagent is correct.
Okay, why I say here, confirm if this QC reagent is correct. It contains, if the reagent is the right type, because we have two types. One is RFP, used in the BF700 and BF700 and BF710, because the previous two flow cytometer only got two lasers that lower channels. And here we use the RFPS. Here's the S. You see here 3 laser, because it used in the BF730, the highest configuration one, or the newest one. And also you need to check if the chef life, and this RFPS, these QC reagent need to be placed in the refrigerator. So if you leave the QC in the room temperature, it may not work anymore.
After mixing it upside down many times, drop 5 drops into the flow tube. Enter the quality control interface. After confirming the tube position, the position of the QC can be modified. Place the QC in the corresponding position. Click Auto QC to perform quality control test. If the quality control passes, it will show pass and a green check mark symbol. You can view the data details in the quality control report.
So, in the real life, we may get the complaint from our customer that the QC did not pass. So, in the most of times, you need to, I mean, as a after sales personnel, you need to ask our customer to take a picture of this interface, the QC report.
Exit the quality control interface for sample testing. In the shutdown menu, select the shutdown button. Select shutdown. Follow the prompts. Take out the probe cleaner. Draw 2 milliliters of probe cleaning solution into the tube. And put it in the first position of the plate. The entire shutdown and cleaning process takes about 25 minutes. It takes a long time, so after adding the probe cleaner, you can close the software and computer after clicking OK. Don't wait for the instrument to be clean. The instrument will automatically turn off after cleaning.
Okay, is that clear?
Yes, it's clear for me.
Yes.
Okay, and do you know Chuck has a YouTube website, not website, YouTube account, and he already put this video, and the next video I'll show you later to in his account. So if you want to get this video, you can check his account. I don't know if you know that. And then the next video is the whole process of the sample detection here. Also, I use the TBNK item as an example.
PBNK lymphocyte subsets detection. Sample preparation. Reagents and experimental equipment that need to be prepared. Pipette. Pipette tips, Vortexer, TBNK 6-color antibody reagent. Lyes. Flow tubes and fresh blood samples.
Oh, the blood sample should be EDTA anticoagulant.
The reagent needs to be mixed well before the sample addition. Numbering the flow tubes. Use a 20 microliter pipette to aspirate 20 microliter of TBNK antibody for each sample. The reagent needs to be added to the bottom of the flow tube. Use a 100 microliter pipette to add 50 microliter of the mixed blood sample into the flow tube. Invert the blood tube up and down to thoroughly mix the blood. Pipette 50 microliter of the blood sample to the bottom of the flow tube. Vortex the flow tube to mix the antibody with the blood sample. After mixing, the sample needs to be protected from light and incubated for 20 minutes. After incubation, red blood cells need to be lysed. Mix the lyes thoroughly. Using a 1000 microliter pipette, add 450 microliter of lyes to each sample tube.
Okay, I'd like to notice here, we in this video we used different kinds of pipettes, right? And in the customer's lab, they may do not get so many types of pipettes. The principle is you can see the instructions, of the test reagents. The instruction of the test reagent will tell you how many reagents you need to add, and how many samples you need to add, and how many lyes you need to use, and just notice that number and find the pipette, and on the pipette the wall has the maximum and minimum width. Is like you have to make sure the volume, the each no matter the reagents or sample or the lyes, the volume are within the width.
Add the lyes to the bottom of the flow tube. Vortex the sample to mix thoroughly. Incubate in the dark for 10 minutes. After incubation is complete, the sample can be loaded onto the instrument for detection. Load and run. Use the preload template for machine testing. Select template. Select or create an experimental data folder. You can change the experiment name here. Edit sample information and sample location. Select the record and click Run. The instrument will automatically stop detecting after the stop conditions are met. When the record button is yellow, the instrument will automatically stop recording based on the stop condition. If not, the instrument will continue recording and will not automatically stop detection. Fine-tune the gate position so that it fully fits the current sample. After confirming that there is no problem with the gate, you can wait until the instrument stops. Or you can click Auto Record, and the instrument will automatically test the samples in sequence. Load and run for engineer. Create detection pattern. Start by creating a new.
Sorry to interrupt. Here we see we have
Incubate in the dark for 10 minutes. After people can be select temp.
Okay. Here we see load and run for daily use. I made it here that want to express the following operation is for the operator, not operator, the customer for daily use. You see,
use the preload template for machine.
It starts from open the template here.
Testing. Select template. Select.
And open the template, select the template, and added the name of the experiment, and put the experiment in some files you want to put it in. So after this,
select or create an experimental data folder. You can change.
Here, we now have to add the sample here to make the machine know how many sample and in which place, which position here. And after that, we can click run or auto record. Auto record is highly recommended because auto record means you can detect these samples one by one automatically. Here's the stop condition. So here these are the samples. If we click auto record here, and the samples will be detected one by one. And after the one sample meets the stop condition may be the events of the gate here. We choose to gather these accounts of the lymphocytes, and or we have time limitation. For example, for each sample, we want to detect 200 seconds. And we have the third way, the volume here. For example, if we want to detect 100 microliter here. After the sample detection meets the stop condition, the next sample will be detected, and then the third, the fourth. So test the samples automatically. And okay. And here, load and run for engineer. This is the steps or the procedure how we create the template for our customer. Is how we preload the template for our customer. For the previous one, the customer starts from open the template. So for the engineer, the following video will show you how we preload the template for our customer.
Engineer, create detection pattern. Start by creating a new detection template. File new experiment. Select the desired channel. Select or create a new experiment save path.
The channel you need to select will show in the panel or in the reagents instruction.
You can change the experiment name here. Add new test tube. Delete test tube. Batch new test tubes.
That's 3 ways for edit the sample.
Batch deletion of test tubes. Modify the name of the experiment. Modify the experiment ID. Modify the position of the test tube. Right-click to set label. Set up fluorescein labels according to the experimental instructions.
Okay, this step is to let the machine know which channel we use. Which channel we use to detect which marker. As I said previously, the reagent we use to stain a cell have 2 main components. One component is the antibody or something combined with the marker. And the other component is the fluorochrome, which can be detected by the machine or be acetated by the laser. So we can view this as the fluorochrome and the label as the antibody. So this step is to make the machine know which channel to detect which antibody.
Create scatter plots or pseudocolor plots, according to the graphical template given on.
The purpose of the set up the detection pattern is to set these figures or the parameters as the standard one. For example, for normal test items, this one is TBNK. This figure will be showed in the antibody instruction.
The standard experimental operation. Set x-axis and y-axis. Apply different door shapes based on the reference template. Double-click to finish gatting. Name the gate after gatting.
You see, in the previous moments, there are standard graph or standard results. We can just do the same thing as to make sure we can get the standard plot here.
Right-click the gate to edit its name, color, and other settings. You can also click here to change the gate's color and layer. Select Logic Gate. This means selecting which gate the cells used as the data source for this plot come from. Set the target cell population and stop conditions. Run the test after placing the sample in the corresponding location. Use the slow speed while adjusting parameters. Adjust gain and threshold in real time based on particle cluster position. Increase the gain value, and the cell population in the plot will shift toward higher value. Increase the gain value, and the cell population in the plot will shift toward. Adjust gain and threshold in real time, placing the sample in the corresponding location. Use the slow speed while.
Okay, here the sample rate, we use the slow speed because this step is to find the optimized parameters. So it may very long. It may cause a long time. So we have to make slow rate for in case the samples be ran all of them.
Adjusting parameters. Adjust gain and threshold in real time based on particle cluster position. Increase the gain value, and the cell population in the plot will shift toward higher values on the coordinate axes. First, focus on the forward scatter and side scatter plot. Adjust SEC and SSC gain. Adjust the cell population to be centered and properly expanded on the plot, ensuring it is not clipped.
Okay, I know it will be hard for you, and I will explain this more specifically. Here we see the standard plot like this. So in the video, we are trying to set the plot just as the standard plot. So we see in the previous procedure, we already add the plot and set the here the ASICs, select the ASICs and choose the gate here. And then after setting the form, we are run the machine in slow speed now to get the result. So here we already get some results, but it's not as same as the it's not as same as, sorry, it has not, as the plot we want. So oh, so we see in the first plot, we have to make sure all the cells can be detected, because if the cells like this, the cells be cut out. So make sure all the cells are be detected. And then we see the all events plot just like this one. And we see there's differences between this one and this one. So we're trying to make the real data as this one. So see. The first step is to optimize the gain value. Gain value is like amplifier. It's like the data we detected by the machine, and we have to amplify the data, and the higher the gain, the higher the amplify. So just as the video showed, in this plot, the higher the gain value, you see the x-axis is CD45. CD45 is here. Okay. If we increase the number as 800 or even much higher, the cells will get the righter place, like here. And mean, you see, the higher the gain, the more positive or more higher the signal here.
Next, focus on the all events plot. Adjust the plot so that all cells fall approximately between 10.2 and 10.5.
Here, 10.2 and 10.5. You need now to remember these this number, because you can see that in this plot, 2 and 5, 5, 6, and
and the lymphocyte cluster is clearly separated. Adjust the CD45 channel threshold. Cut off the excess debris. Adjust CD45 channel's gain. Let the limb cluster around 10 to the 4th power.
Again, you need now to remember these number, like the 10 to the 4th power, because in the flow cytometer, the key point is to make sure the cell groups are easily to classify these groups. So as I said, show well boundaries against the axis. So we recommend, or we in the optimized, or the theoretically, we need the cells locates to 10 to the 2nd power, or 10 to the 4, 10 to the 5. But actually, in real life, as long as you can make sure the plot is like this, and make sure the cells, or for example, here's the lymphocyte, the cell groups can be easily gate, the gate here to gate. That is okay.
Adjust the FIC channel. Let the CD3 cluster be around 10 to the 4th power. Adjust P channel. Let the CD3 CD4 positive cluster be around 10 to the 4th power.
So again, you need not to remember that number. Why I showed number here? Because this is the training video. I want to make these are standard. You see, these group of cells are well separated and easily to gate.
In the same way, the CD8 positive particle cluster of the APC channel.
So you see why it decrease the number here.
Positive cluster be around 10 to the 4th power.
Okay.
In the same way.
Okay, here we see in this figure, in this plot, the CD3 is okay, because it's well separated, and the positive groups are actually locates good here. But we see the y-axis here. The positive groups are too high. So we need to make these groups down. So as I said before, the higher the gain, the more positive the cells will be located. So for the y-axis, the more positive means it will go up. So but now we need these groups be down. So we need to decrease the number. You see?
The CD8 positive particle cluster of the APC channel.
Here's 700, and now he's changed to down.
Convert the CD19 particle cluster of the APC channel to around 10 to the fourth power. NK positive particle clusters of P channels. Adjust to around 10 to the fourth power. After the gain is adjusted, you can click to stop running for compensation.
Okay, here's we optimize the gain value, and we see the plot is not very good because you see here's like this. Why it gets showed like this? Because the compensation, because the compensation. Sorry, I think I neglect talk the compensation here. You see, these are the channels, the channels of the emission spectrum of each fluorochrome, the sphere, the ball, the fluorochrome, and we see, for example, this two, the red two. We split the signals by a series of filters, right? And if these yeah. Okay. If this is the final spectrum that we detect, we see in this channel or in this width, in this width, not only the PE series, not only this one, the target fluorochrome can be detected. There are also some signals that skipped from this one, AF700. So it means even although we define this width signals as the PCS 5.5 channel, but in actual, not only the signals from this fluorochrome be detected, but also some signals skipped from this fluorochrome. So we need to minus these signals, the signals skipped from the AF700. So that means we need to do the compensation.
Adjustment compensation can also be adjusted in the running state. Load and run for engineer. Compensation adjustment. Create a new plot.
Okay, again, the main purpose or the purpose of the flow cytometer is to classify or to gate this is the gate to gate the cells easily. And now there are compensations. So maybe here we see if we want to get these PE positive signals, but you see here's not right. This is target cells, but the PE positive signals are not only comes from the target cells, but also the other cells. So we have to make sure make these group of cells down and make sure the PE positive information or signals all comes from the target cells. So we now open or set a new plot to show the compensation of each channel.
According to the standard graph to operate.
You see, this is a target plot, the standard plot.
First, select a y-axis channel. Modify the x-axis channels one by one.
So because we have so many these channels, 6 channels, and each of them may have compensation. For beginners, I don't want you to remember which channel has the most compensation with which channel and which one should minus which one. That is complex to remember. But here we use the simple way, but takes longer time. It means we fix the y-axis and change the x-axis to show if there are compensation.
Observe which particles are not clustered or floats linearly to one side. Click the Auto Compensation button. Ensure the particles form clusters and do not drift toward one side. Pull the adjustment lines above and to the right of the graphic. Clusters change in real time.
So how we say the compensation is right is like, for example, in this plot, we know these 2 groups of even it shows not 2, but mean 2 groups here and here. These group cells are this channel negative, CD8 negative. So the line connecting the center of these 2 groups should both negative and show both the same position of the y-axis. It means the line connecting the 2 groups should parallel to the x-axis, like this.
Tails of the compensation value below will also change accordingly. Adjust while observing the differences between the scatter plot above and the standard plot. Replace the horizontal and vertical channel. Continue to adjust. Make the particles cluster together without drifting to one side. This compensation adjustment method is suitable for operators with some basic experience.
Okay, it means we fix one axis here and then change the x-axis one by one. It's suitable for some operators who have some experiences, because in that time you will know which one has compensation with which one. So maybe, for example, we know 2 couples or 2 groups have the compensation. We directly change the compensation of that groups. And
Quick adjustments when you already know which related channels require compensation. Load and run for engineer. Compensation adjustment.
This compensation method is for the beginners because
You can also build scatter plots and batches. Put out all the graphics. Select all fluorescence channels for both the X and Y axes.
here we choose to put all the figures out with the okay, all the figures out. And
Long press shift and select all graphics with the mouse.
and then change the
Right click and select
Topic.
batch graph attribute settings. Select logic gate as lim. First, use the reference plot to determine the positive and negative distribution of cell populations in each scatter plot. Then adjust the compensation. In each plot, the center of the x-axis negative population should be aligned at the same x-axis position. Alternatively, the line connecting the centers of the x-axis negative populations should be parallel to the y-axis. The same applies to the y-axis.
So in this way, you just put all the figures out. So you see maybe if this group like this and this group like this, you directly drag this figures and show it in the whole system.
After adjusting, you need to delete the excess pictures. Press delete. The position of the crossgate can be adjusted. Make graphics look better and more beautiful.
In this step, I want to show that even though the parameters is good, but you know the axis can be adjusted too. For example, you see here the axis is like this, but here we show too much about the 0 to the the number below 0 to the 10 to the third power. So the signals is like skipped out of the plots. We have to make it better. And remember the main purpose of the flow cytometer is to make the groups easily to gate or easily to classified. So we need to drag the SX2 to make sure the plot separate enough, have well boundaries.
A good flow cytometry scatter plot should be as centered, symmetrical, and evenly distributed as possible. Load and run for engineer. Set up the statistics table. Click the statistics table button. Right-click and select statistical settings. Select the groups you need.
And this step you need
Volumetric absolute counting is only
This step you need now to remember too. You just directly ask the doctor which population you want to see. Which the status information of the population you want to see. Make the doctor select.
applicable to samples that are not centrifuged with the supernatant discarded.
Okay, this concerns another topic. You know, we can do this cell accounts, get the cell account information right. It's like two ways. Just one way is like the hematology analyzer. Directly gets the volumes. And now you have to put the dilution, sample dilution.
You need to enter the dilution ratio. Dilution ratio equals total liquid volume in the tube divided.
Okay. If you do not discard any liquid, the dilution ratio will be the total volume in this tube, divided by the sample volume you add. For example, after preparation, the total volume will be 1 milliliter, but we only used 50 microliter of sample. Then the dilution will be like that: 1 milliliter divided by the 50 microliter.
By sample volume. Statistics form support simple calculations.
Okay, you need to remember this too. Just
Right click and select
stop.
export. Report viewing and LIS settings. Report viewing. This is the default template. Custom templates are supported. LIS setting. Click setting. LIS setting. Open the URIT LIS system. Log in directly without password. Confirm that the selection is enabled in the LIS settings. Send icon changes to the selectable state, showing that LIS is connected. Select the report to be sent. Thank you.
Okay. Last part is the sample reports and the LS connecting. I think you are more know more things than me, so just skip this. Okay, I think today's training is finished. Do you have questions, or if you think the application operation is too complex, that is okay, because almost no one can handle that in one training, and just have the basic knowledge and the steps, why we should the operation steps, and why we should do these steps. And the order of the steps first is to, for example, change the gain value and then the compensation. And just do make the plots or the results as same as the standard plot given by the test item or reagents instructions. And do you have questions?
Just a thing, Clara, for the software,
Yes.
I think we would like to have like a more kind of comprehensive knowledge of the software. I mean, that was my idea. Like, for
Okay.
example, usually when we have to do the training and the installation for the instruments, we have to
Okay.
explain to the user what it is
Okay.
in each menu. For example, you have a file, template, cytometer settings, and
Mhm.
in each of those, we will have some options, right?
Okay.
So,
Okay.
yeah,
Okay, I get it. There's a guidance of the of each the software button means like this. Show the connection, the procedure and the bottom usage like this. I will
okay,
send this file to you after this training, and because I already installed the software in my computer, I can show it now too. So in the normal or daily operation, uh, reopen it, I think. Here's the Sator, here's the software. Click, double click, and then okay. Here's username, admin, and password 12345 6. And then this one is the guidance actually, but normally we do the QC first here. Zach, you see, because I haven't connect any machine now, cannot do that. See, just prepare the QC and then put the QC to the first position and click Auto QC. Then after the QC, we close the QC here, and then for the operator or for the customer, the only like templates, choose the templates, or it's better, actually it's better to here, to click here. It's more easy to get the information. It's like this. Here's the TBNK. Okay. Like this, the templates already be preloaded like this. And let me introduce each bottom means. First, I want to introduce the tube here, tube bar. This one is add tube. Add tube, you see, add one tube, add tube. And this one is to add multiple tubes. For example, if I want to add 4. And this one is to delete. Delete. This one is delete multiple tubes. Select all, or just leave one. Here to open the folder. Open the clip. It means it will show you where you store or put the data. And then the compensation matrix here. Optimization matrix. And empty clear all position. It means, for example, if I have 5 tubes, in total there will be 6, as we add 5 more. And you see the position will like this: 1, 2, 3, 4, 5. And if I do not want to detect these, I want to skip the first one. But sorry, the first one, and we can empty the position. And then here, click this sample. This one is auto set to position. It's like this to let the machine know I put the samples from here, and the location will be the first, the second, and this. And this one is to show the procedure of the loader or the sample. Unassign and then like this. And then the and then here the plots histogram is like this, the count. And then add a multiple histograms like here. Then plots. This is dot plot. Add multiple dot plots is like, if I want to see the CD3 and the y-axis one I want to see is CD4 and CD8. You see, click CD3 and CD4 and CD8. It will have this like CD3 and CD4 and CD8. Okay, the procedure, these 3 do not commonly used. And here we open a table here. And here see the tree diagram means where the gate comes from. Actually, you see here we see the P1 here comes from all events, and the lymph side comes from the P1. Means this plots, these signals comes from P1 gates, and in the P1 gates we classified or we gate the lymphocyte. And in the lymphocyte here, in the lymphocyte gate, we gate the CD3. So diagram like here. CD3 comes from lymphocyte comes from P1. And add means you can add 2 groups together if the doctor needs. Yes. And then this one, these are different shape of gates. For example, this one is used in the histogram. So you see, you cannot choose the gate here, but you can choose here. It's like here. And this one do not use that often, so skip that. And this one is like this. Click, 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 double click. Okay. And we can add the name here, for example, LYM1 here. And this one, triangle ones, like, and then other different shapes of gates, like no. Mhm. Mhm. Okay. Okay, this is like and then here's the color. Sorry, the logic dot means you can add or delete some, combine two groups together. And this one is the color and layer of the cell groups. For example, I add too many. Here, CD3 and CD4, we can choose the color. And zoom in, zoom out. Because my computer is not suitable for this software, there are actually more. Cannot show it. And I can open the experiments that have the already have the information. Okay, I think here. Okay, this one already get the information.什么什么调? Okay. For example, add tube. Add these tubes, and then I want to add the plots. This one here. It will has the data here, and normally we use the input side. Okay, and in this plot we can choose a gate like this, L6. If this one is the cells or the information I'm most concerned about, so I can do this here, the layer and the color. You see, the gate means L6, or you can add the name as you want. Most, just like my name here, okay. Okay, my name here. And then I want to see this is like make it like this. And if oh no, so last time I okay. You see this group of cells, my name here, is the cells we most concerned about. So I set the color and then put it into the first layer. So in the rest plot, this group of cells, the purple one, will show as the first layer. Will make you notice this group of cells in each plot. if I hire this one, it will different. It's like this. And here we can see the status data here. And also the tree diagram here. And these are the tube and the gaze and the plot. Okay, here's the I like to call it the real data or the real situation of the detection. For example, flow rates now is like this. When running the data, the number here will change. And this is the flow rate, just as I introduced in the video. In the daily use, we choose medium, but when we optimize or set the parameters, we choose slow. Then stop conditions. You can ask the doctor to define these. It means the stop condition means when record the data or you hear or record or auto record. When record the data, after the detection or the information data gathering meets the stop condition, the detection will automatically stop. Okay. Acquire settings is where we open the gain value interface or threshold. Threshold is like here we choose set I cannot add it here, but I can add it here. Haven't be detected, so the detection or the acquire parameters can be changed. But for samples already be detected or record the data, the detection parameter cannot be changed. So we can change the threshold to cut the debris here. And the width, it's like, do not use that often. Okay, this is the first mean interface actually. And the file means that you see new experiment. Well, you set the experiment from the beginning. The first thing is to select the channel, just as introduced in the video. New plan experiments, that is complex usage will not introduce in this training. And conversation, another deeper information. And new experiment from templates means actually the same as here. Open a new set, start detection from template. And then open experiments, just as what I did before, just like open the previous data. And then save this data. Save as means save as a new name or locate or put it in a new place. And save as template. Here, save as template is a key function, means, for example, my name here. After save as templates, we see here, these parameters will be fixed and be loaded at the template. And the next time the operator can open it here. So it's commonly be used in when we customize these parameters, or the test item for the customer. We set the experiment, optimize the parameters, and how we preload the templates use save as template here. And import FCS file. FCS file is a standard file for flow cytometry data. FCS files can be analyzed not only by this software, the Sator, the UR software, but also by the Beckman software, BD software, because it's the standard data form. And recent data is where you can click recent data if it's easier to open the previous data. And then the compensation experiment, recent template. Okay. And here's the experiment templates as I told before for operator for our customer. The daily operation will the daily detection will start from open the templates here. And the cytometer will acquire sightings is like here. Basically, these two are the same bottom. And here we also used some other functions like remove bubbles to remove the bubbles of the liquid system, cleaning, deep-cleaning, and drag the sample leading and so on. Sample injection mode, we have 3 injection modes like aerosol is a 2 plate and a macro plate and the single tube. Maybe normally will not change after we deliver the machine to the customer, but for distributors, they may need that function. And the set label here is to when we set the template from the beginning. And here's to make the machine know which channel we stain or we detect which marker. And these settings, even display settings, is like normally do not use that, need not to handle that this time. Consumable management and options is like some engineer thing. I don't know actually. Then language setting, and then user management, account management, and then QC is like already been. Then maintenance needs password. The password be changed once. So let me check. ENG1. Okay. When you enter the passwords like this, there are many passwords result in many functions. And the fault center is like record is the log recording, and shut down because I do not connect any machines my computer, so cannot shut down. Help file and others. And I think here's another thing I need to tell you that, any machine will have some errors, For example, there is a prompt that's error E01 or something else that your which parts have which error and so on. So normally the prompts has the SWIP, I don't know how to say, sample here. So you can click the sample, it's like SWIP. You can click here and the machine will fix that error automatically. normally normally you can deal with the error this way. But in some hardware error or some complex error, you need the engineers need to come to the customer or something else. Okay, I think okay. Here's the communication means whether you already connect the machine to the computer, and operation means the okay. This is the situation or the status of the machine and the computer. And the carousel shows now this instrument are configured with the two plates, the carousel plate. You'll see here we can we have not only carousel but also single tube and microplate like that. Okay, I think that is clear. Is that okay?
Yes, Clara, thank you very much. It gives us a better look of the software. I think we have
Thank you.
a little bit curious about the rest of the things because we didn't see that yet.
You want to learn more about the installation and maintenance? Actually, it's not my part. It's not my job, and I'm not very good at it. I can only do the basic installation and maintenance. So it's better for the after sales department to organize the training, I think.
Yes, thank you. You mentioned me that you have some PPTs and documents for that also. Yes. If you can share to us, please, so
Yeah.
we can take a look of that and
Yes, of course. The data the files I mentioned, like the TBNK operation guidance and the after service manual or some installation guidance or some just these things I will share them with you.
okay.
Okay, maybe 2 hours later because it's lunchtime actually.
Yes, yes, it's okay. Don't worry.
Okay.
Thank you very
you
much,
for
Clara.
your
Thank you for
time.
your time.
Thank you. Thank you. If
Thank
you
you
have
very
other
much.
questions,
Thank you, Clara.
if you have other questions, you can
Thank you, Clara.
call me or do okay. Okay, thank you for your time. I hope you learned something. I hope
Yes,
I
of course.
haven't with yourself. Okay, thank you. Bye bye.
Right, thank
Bye
you.
bye.
Bye
Bye.
bye.
Thank you.
Bye bye.